Let's talk muscle function. All right, now function of the muscle. We are really going to focus in on contraction. There's other functions that muscle does do, whichever. For example, muscles keep you warm. When you get cold, you shiver, and that shivering generates heat, or it transforms chemical energy to heat energy technically, and that warms us up. We'll talk about the heat generated through muscle contraction during exercise as well. But the muscle can also protect the different parts of the body. So it can protect internal organs and regulate core temperature. But really what we're interested in is the primary function of muscle as related to human movement, and that is to produce movement, maintain posture, stabilize joints. In essence, the muscle function that we're interested in is the ability of the muscle to generate force. Now remember, the muscle is the contractile portion of the myotendinous unit. The muscle connects to the tendon via all the connective tissue that we talked about in the previous presentation on structure. But nevertheless, we were interested in the contractile component of the muscle in vivo, in the body. So let's talk about that, muscle contraction. Well, okay, so how does a muscle contract? Well, we're going to go through it briefly. You would have gone through this in a few different classes already. We have a central nervous system signal, CNS. This is just a way to start it. And we have a transmission of the signal from the brain to the motor end plate via alpha motor neurons. It's electronic signal, electrical signal. We actually measure that electrical signal by using EMG. Not of the nerve at this point, so I'm a little early in mentioning that. But upon reaching the postsynaptic membrane threshold, a signal is transmitted throughout the muscle fibers of the motor unit. And remember, motor unit is the alpha motor neuron and all the innervated muscle fibers. Remember, muscle fiber is muscle cell. So a motor nerve, a motor neuron can innervate many motor fibers, or a few. If we're looking at dexterity, how the fingers work, there's going to be a lower number of muscle fibers per motor unit or within a motor unit. But if we're looking at big muscles like the rectus femoris, the quadriceps, the vastus lateralis, and so forth, the gastroc, and the gluteus, those are big muscles. Those are going to have lots of muscle fibers per motor unit. So motor unit is the nerve and all the muscle fibers that it innervates. All right, so muscle contraction. The model that we use to understand muscle contraction is called the sliding filament model. The filament that is being referred to here is the myofilament, the thin and the thick filaments, the actin and the myosin filaments. And in essence, they slide over each other. And that's the model that we're going to talk about here. So muscle shortening occurs due to the movement of the actin filament over the myosin filament, formation of cross bridges from the myosin filament between actin and myosin filaments forms the power stroke, and that pulls the actin filament towards the center of the sarcomere. And there's a reduction in distance between the Z discs or Z lines of the sarcomere, and the sarcomere shortens. And remember, the sarcomere is the functional unit of a muscle cell, of a muscle fiber. It's the unit that we use to explain muscle contraction. All right, let's look at this from a cartoon perspective. This is a sarcomere from this Z line to this Z line. Here's another sarcomere from here to here. Is the M line, the middle of the sarcomere where the myosin filaments are anchored. This is the myosin filament. This is a thicker filament. This is the thin filament, the actin filaments. And these protrusions are the cross bridges, the myosin heads that will actually contract connect with interact with the thin filament, and that is a cross bridge. Here's our A band, the darker region of the sarcomere, and the I band, the lighter region of the sarcomere. Well, this is crossing two sarcomeres, but that's okay. The lighter region of the muscle fiber. I shouldn't say the lighter region of the sarcomere. Sarcomere is from Z line to Z line. All right, so as we contract, as we shorten, as we see this sarcomere shorten and this sarcomere shorten here, okay, picture 4 is shorter than 1. We have a contraction. We actually see a reduction in the light band of the muscle fiber. So the light band gets smaller as the muscle contracts. The A band actually stays the same. And because the A band is really defined by the length of the myosin protein, the myosin filaments. And that A band is going to stay the same length. Okay. All right, so let's look at this a little bit more detailed. Here's the thick filament here. These are a bunch of myosin proteins here. And here is, this is called the myosin filament in total. This is a myosin protein. Here's the myosin head, myosin tail. And they're all linked together to form the myosin filament or the thick filament. This is the thin filament. This is a bunch of actin. This is a bunch of actin right here. Here's another part of the thin filament. This long band here. This is tropomyosin. And then there's one more part of the thin filament, that's troponin. So the troponin actually blocks, the troponin will bind ultimately with calcium. And when calcium binds with troponin, it will change where this tropomyosin is located on the thin filament. And when it changes where the tropomyosin is located on the thin filament, it actually exposes a portion of the thin filament, the actin portion, that interacts with the myosin head. So without that calcium binding with troponin, there's no potential for the myosin to interact with the thin filament. Okay, and that's an important part. We're going to go through this and again in another slide. This is in your textbook, figure 1.5, chapter 1. We can start anywhere because this is a cyclical process. And in this cartoon, we have a myosin head, which is connected to the myosin filament, not really shown here. And then we have the thin filament, which has the actin, which are these circular proteins. We have the troponin, this part right here, which is still part of the thin filament. And then we have the tropomyosin, this longer protein that runs along with the actin. Again, we can start anywhere because it's cyclical, but we'll start here at number 1. There's the resting state. There's no interaction between the myosin head and the actin. There's no interaction between the myosin head and the thin filament. But calcium arrives after a signal from the nervous system, goes along the cell wall of the muscle cell, the sarcolemma, and then it goes down into the transverse tubules. And that calcium in the transverse tubules, that motor signal in the transverse tubules causes calcium to be released from the sarcoplasmic reticulum. That calcium then goes inside the muscle and interacts with troponin. When calcium interacts with troponin, it changes where this tropomyosin is located on this thin filament. And as that myosin changes where it's located, there's an active site on the actin filament that interacts with the myosin head. This is just showing that adenosine diphosphate and inorganic phosphate are located on the myosin head already at this point. Okay, so the myosin head is actually in a high energy state at this point. meaning it's cocked and ready to shorten. It's ready to, it's ready to change its shape. Okay, that's where we are right now. We go here, the myosin head attaches to the actin binding site. When that happens, the myosin head flexes. It changes its shape from this position to this position. And as such, because it's bound to the actin, it pulls the thin filament towards the center of the sarcomere, towards the M line. Adenosine actually it releases inorganic phosphate at this stage, and adenosine diphosphate. And now we have rigor. Okay, we have the myosin head is now in a flexed state, and it's stuck there. And it will stay stuck there unless ATP comes along, adenosine triphosphate. And when adenosine triphosphate becomes available, it detaches the myosin head from the actin portion of the thin filament, and it cocks, it puts the myosin head back in a high energy state, ready to change shape again. All right, so that's where we're up here. So ATP, adenosine triphosphate, is split to adenosine diphosphate and inorganic phosphate, and it goes, the myosin head goes back to a high energy state, ready to interact with the thin filament again. Calcium comes in, myosin head binds with the thin filament. It changes the shape of the myosin head, this portion right here. It pulls the thin filament towards the M line, towards the center of the sarcomere, causing a contraction. And then it releases again if ATP is available. If you die and you do not have generation of ATP, you will have rigor mortis. That means rigor, contraction, mortis, you're dead. So the muscle tightens and it stays tightened at that point, because it needs the ATP to release the myosin head from the thin filament and put the myosin head into a high energy state ready to contract again. This cyclical process just happens and happens and happens and happens. Happens really fast, really fast. As long as calcium's arrived, as long as there's a motor signal being sent to the muscle cell telling calcium to be released into the muscle. All right, so now a question that Noakes raises, is the myosin binding to actin firm or a series of loose couplings? Because ultimately we need to understand eccentric contractions. I know you know what eccentric means, is your muscle's trying to contract, but it's actually lengthening. And there's a bit of discussion out there in terms of whether that eccentric contraction is breaking of the myosin connection to actin, or if it's just a loose binding and it slides, and the myosin head slides over the actin as the actin gets pulled away from the M line. So this is an interesting discussion, and we'll talk more about this later on when we talk about endurance performance, especially with running. All right, so the energy for muscle contraction, we talk about being adenosine triphosphate. And we say it's required for muscle contraction, but really the adenosine triphosphate is really to re-energize, to put the myosin head in a high energy state. Myosin ATPase is what breaks down ATP as the muscle fiber contracts. Sources of ATP, phosphocreatine, glycolysis, oxidative phosphorylation. These are our energy pathways. And we'll talk about each of these as we go along. But these energy pathways are all used to describe generation of ATP, which is then used in that cyclical model of muscle contraction to re-energize, put the myosin head in a high energy state so that once it does bind with the thin filament, it pulls it towards the center of the sarcomere. Okay, muscle role of, muscle function, role of muscle. We have different ways to describe the role that muscle does when it generates a force. we will say agonist as a prime mover. And when the muscle is generating force to cause a movement. So if I'm doing elbow flexion, the bicep brachii is an agonist. An antagonist contracts to resist movement. So if I'm doing elbow flexion, maybe that my triceps acts as an antagonist to resist elbow flexion. Muscle can also generate force to stabilize. Function, in that case, it fixates an area so another movement can occur. So if I'm swimming, maybe I'm trying to stabilize my scapula in a way to allow the humerus to move within the glenohumeral joint. Muscle can also play a role as a synergist. That means it's going to assist another muscle. All right, so maybe not besides the bicep brachii, I've got some other muscles within the arm to assist with elbow flexion. And then it can also be a neutralizer. When the bicep brachii contracts, it also tends to cause supination of the forearm. And we have muscles that will contract to resist that supination. And therefore, those muscles would be called, classified as acting to neutralize of movement that we don't want. Muscle action, isometric, no change in muscle length. This gets a little tricky as we go through this material. Typically, we say isometric contraction is happening when we do not see any flexion or extension, for example, at a joint. And the trick is that the muscle, the muscle is connected to tendon, which is elastic tissue. And so the isometric action is really describing what's happening at the sarcomere level, but we are often guilty of applying that to a muscle tendon unit, and then we can actually have some muscle sarcomere shortening. It gets, sorry, I'm doing a bad job of explaining this. Let me get to concentric and eccentric. I'll try again here. So isometric, no change in length of the muscle. Isotonic is a change in length where we're using the same tension or same external weight. So this would be free weights. Concentric, muscle shortens during contraction. Eccentric, muscle length increases during contraction. And what I was trying to explain was, let's say if we take running, for example, and our foot strikes the ground, we tend to say the knee flexes, and we see that, and we can quantify that, we can measure that. And we might say that the muscle of the quadriceps is acting eccentrically. But interestingly, the muscle itself, the contractile unit of the muscle tendon unit, may not be changing length at all. And the length of the muscle tendon may be getting longer only because the tendon is stretching, the elastic material stretching. That's what I was trying to explain before. So we are guilty of sometimes mixing these terms up, but in essence, isometric is no change in muscle length, concentric is shortening of the muscle, and eccentric is lengthening. There's just some intricate details in there when we start looking at human movement. Isokinetic is another term that we'll come across. Same kinetics, same force. This is a tricky term, but in biomechanics, hopefully you covered it. This is where angular speed is constant during contraction. And in order for that to happen, you need a variable amount of force being applied to an extremity. Isokinetics can be concentric or eccentric, where speed is constant. And you pretty much need a special machine to be able to do this. Biodex, Cybex, hydraulic machines, things like that. Okay, so this is part 2 of chapter 1, sliding filament theory. You should know that, role of muscle and muscle action. All right, thank you.